Hi, this is Miss Litton, and these are my wonderful AP biologists. Say hi. Hi. Okay, and so we're reviewing Unit 4, and this kind of has a modified review. You can log in, but you'll see. It captured, like before we had where you would log in via the computer. I used to use clickers. Do you remember clickers? And so it captured some of the review questions like this so you can look at them. So you'll see them, but I don't think you can answer them until I'm on a later slide. So it's kind of an amalgamation. Starting with chapter 16. Okay, I'm kind of out of order here. The first one, we look at chapter 16. Oh, hi guys. Thank you. Okay, let me go right here. Okay. Okay, there we go. Could you answer that question? What one is that one? Yeah, when you only have a few individuals surviving of unfavorable times. When you have unfavorable times, it's always a bottleneck. Okay, and on that note, I want to talk a little bit about the big ticket items on chapter 16 that you're going to need to basically know. You're going to want to know that, and this is kind of also applying to your essay portion, though we'll focus more on those essays when it comes closer to the final. But you want to definitely know what the definition of evolution is, and you want to be able to differentiate between microevolution and macroevolution. What would you say microevolution is? Okay. What is microevolution versus macroevolution? Microevolution is be better within a species. Yeah, it's within a species. Whereas macroevolution, okay, sorry. Okay, reconnected. Microevolution is within a species. Macroevolution would be to an entirely different species. We talked about that at the very beginning of chapter 16. And then we talked about that evolution, in essence, is a change in what kind of frequency? Allele frequency. So anything that changes the allele frequency is going to be a type of evolution. If you're still the same species, then that's micro. Now, the whole definition of a species thing, we're going to get into that in a later chapter and what are some of the problems with that. But when we talk about things that could change allele frequencies, this is when we start talking about Hardy-Weinberg. And you want to make sure you understand the big ticket part of that. And the big ticket is saying, here are the conditions you need to meet in order to maintain those allele frequencies at Hardy-Weinberg equilibrium. Because those 5 conditions are so difficult to maintain, then by default, what has to be happening? Evolution. Why are you being so quiet today? Are you just tired? Okay, you have to be more engaging because I am sick and tired. Okay, so when we think about Hardy-Weinberg, 2 equations should come to mind. And you need to be able to differentiate between what those 2 equations mean, right? Could you do that? Could you compare and contrast those 2 equations and when you would use them? And I'm going to just fix my board. Why don't you talk to the person next to you right now and tell them, how do you know the difference between those 2 equations? Oh, good. Okay, what's the first equation? Okay, P plus Q equals 1. And my board is shifting around, so it's going to drive me crazy. Okay, and what's the second equation?
P squared plus 2PQ plus 2 squared equals 1.
Okay, so when you talk about P and Q, you're saying that there are how many alleles for a particular trait? It's for simplification, but how many alleles? 2. Okay, and the P stands for the dominant allele, and the Q stands for the recessive allele. And when we say you're either going to have big T or you're going to have little t in an allele frequency in your gene pool, right? Here's my gene pool with big T's and little t's floating in it. How this plays out into your population, that's this part right here, the second equation. Because we're diploid organisms, then you have 2 copies of each allele. So you're either going to have 2 P's, or you're going to have a P and a Q, or you're going to have 2 Q's. So if I say the allele frequency was 0.7, the dominant allele frequency was 0.7, could you solve for everything else if I said the dominant allele frequency was 0.7? Okay, so let's think it through. The dominant allele frequency, what am I talking about when I say that? I'm talking about P. P is the dominant allele frequency. I'm not talking about the trait. If I wanted to talk about the trait, I would say 70% showed the dominant trait. That's different. If I say the allele frequency for the dominant allele was 0.7, do you see how that's different? Okay, so you want a different, if I'm talking about any kind of trait, a trait is a diploid organism. Are you tall, short, round, wrinkled, blue eyes, brown eyes? Those are traits. Those take 2 alleles to get. Okay, and that's what I'm talking about, one of those other ones. So if I say the dominant allele is 0.7, I know the recessive allele has got to be what? 0.3. Because 7 plus 3 equals 1. Now, if I know those allele frequencies, I could then predict what I would find. If this is what's in my gene pool, 0.7 and 0.3, that's in my gene pool. How does this play itself out in a population if they are all interbreeding with each other, How would this play out? Well, I would expect how many of them to be homozygous dominant? What? 0.49. And you got that because homozygous dominant is the P squares. So that would be 0.7 times 0.7 would be 0.49. Those are my homozygous dominants for that particular trait. Okay? How many of them would I expect to still show, the board is so messed up right now, how many would I expect to still show the dominant trait, but they would be heterozygotes for it? 0.42, exactly, because I'm going to do my 2PQ. I'm going to do 0.3 times 0.7, 0.3 times 21, times 2 is 42, 42%. Okay, would be this. And how many would be homozygous recessive? 0.09, which would be 9%, right? 0.09 is 9%. So I could have approached it any way. I could have said, like I did for you, I could have said the dominant allele frequency was 0.7, or I could have said 9% showed the recessive trait, or I could have said what? 91% showed the dominant trait. The 91% that showed the dominant trait includes the P squareds and the 2PQs. Okay? The biggest mistake people make is they go, 91% showed the dominant trait, and they square root that and call that P. Right? But you can't, because the dominant trait includes the homozygous dominance and the heterozygous dominance. Okay? You could take the recessive trait, square root it, and get your Q, and then solve for it. You just have to go, 91% show the dominant, therefore 9% show the recessive. I'll square root that, solve for my P, and now work forward, and my P squared is 2PQs. Okay? As long as you don't spend time memorizing, just make sure you make sense. I'm a diploid organism, so I have either 2 P's, a P and a Q, or I have 2 Q's, right? But where we came from, my gene pool, right? I look at the entire population, they throw all their P's and Q's in that gene pool. And look, 70% of them had, of all the alleles in our entire population were 0.7 were dominant, and 30% were recessive. If it stays 0.7, 0.3 in our gene pool, no what is occurring? No evolution is occurring. What kind of things are going to throw off our gene pool? Well, 5 fingers of evolution. What are the 5 things that can throw it off? Small population, because what are they susceptible to? I just asked you a question about it. What are small populations susceptible? What? Bottleneck? What is that a part of? Genetic drift. Drift. If you're a small population, you're going to drift easily. It's not It's not going, nature's not selecting, you're drifting just due to chance, you're so small, any one thing can throw you off. Or, you know, a little wave can tip your buoy, you know, where a big old yacht would stay afloat. You with me on that? Smaller population, more susceptible to genetic drift, okay? And what are the two types of genetic drift? Bottleneck, which means you what? Went through a disaster, a crisis of some sort, pruned you back. A crisis pruned you back. Okay? Or what's the second one? Founders, which means you have a group of individuals starting a whole new colony, and that group of individuals doesn't represent you well. Okay? Founders effect. Okay? That's genetic drift. What's the next one of our 5 fingers? What? What? No, what's this one? Okay, so if you are, you mean non-random? Okay, so non-random mating, where you're choosing this individual over that individual, that could throw off your gene pool, because you could be selecting for those who have the dominant trait or selecting for those who have the recessive trait, right? Okay, and then what's the third one? Mutations. Mutations, you could have the dominant allele mutate into the recessive, and that could throw off allele frequency. Pointer finger, gene flow. We could have 2 gene pools, you know, joined together, right? You could have a whole bunch of people come jump in our pool. That's going to throw off. Or we could have people leave our pool, right? And they're going to take their alleles with them. And then the thumb, not natural selection. Nature selecting one trait over the other. And remember, it's natural selection that can lead to what? Adaptations. Adaptations. Okay? And so we went through, and basically here in chapter 16 that we just finished up, is we talked about each of those, then we talked about them a little bit more, and then we focused on the types of natural selection. So if we look at all of them, we have some where you favor the middle ground. What do you call, what kind of selection is that where you favor the middle ground? Stabilizing. Think I'm stable. Stabilizing selection. I'm very stable. Okay? I could favor one extreme or the other extreme. One, you know, I'm going one direction. What is that called? Directional selection. I kind of gave that away. Okay? And if I favor both extremes, disruptive. Which ones could lead to new species? Disruptive and directional. Good. Stabilizing won't. It'll favor the norm, right? And then we said there's another kind of selection, sexual selection. And we talked about sexual dimorphism, where males and females are different. And we said sometimes there's intrasexual selection and intersexual selection. Interselection is like when a female chooses the male. And do you remember the 2 hypotheses of why a female chooses a male? Good genes, which means they can provide nice, healthy offspring. What was the second one?
Runaway.
Yeah, runaway. They're running away with a trait, okay, that's based on looks. We said if there's intrasexual, male between, males fighting other males to control a group of females or a female, they can what each other? Fight each other, and they establish what? Hierarchies. And whoever's at the top of the hierarchy, there's a cost-benefit analysis, because you're at the top of the hierarchy, you have to do probably more fighting, so you could become what? Injured. And also you have to eat more food because you are larger. Yeah, exactly. So we talked about that type of sexual selection, and we talked about sexual dimorphism, and then we talked about things that maintain diversity. We still have diversity, and that's because traits can be hidden in the, not the homozygote, but the heterozygote. And we said sometimes even a heterozygote can be favored, right? Because it can give you some sort of advantage. Something else we talked about. Oh, you can only, as far as natural selection goes, if there's not, you know, why don't we have like sticky hands and fingers so we can climb walls, okay? Why haven't we evolved that trait? Because that could be helpful, right? But that is not in our genes to come up with, right? Other organisms maybe went that way, a gecko, okay? that they have that, but that we can't just spontaneously get that trait. We're limited by the ingredients and the DNA we can have. That would require several mutations in order to acquire that trait, right? So we're limited by our ingredients. And every trait that we have has a cost benefit. And the example I gave you is, it's a benefit as a trait to be able to be upright, but the downside of that is all the pressure we put on our spine, and we can end up having a bad back. Right. So we talked a little bit about not every trait is perfect. Just because we have it doesn't mean that that is great for our survival. Okay. All right. Okay, that was chapter 16 in a nutshell. And I have a couple other questions on here. Just go through them. Oh my gosh, I'll do them on here. My bad. Sorry. Okay. True or false? It's true. Genetic drift are two kinds of chance. If you're drifting, it's by chance. Think about it. If you set something to drift, it's by chance. It's not like it's got a little engine on it and it's going to something. It's just drifting. It's all due to chance where it goes. And smaller populations are more likely to have that. Maybe it's going to do that every time. I don't want to learn more. Okay. Natural selection occurs. There we go. Do you agree with that? True? Okay. Did you try these? Do you want to try them? I gave you the answers to all of these. I mean, in the presentation, I give you practice problems. Do you want to try it? Do one? Okay. So why don't you see how you would attack it without me helping you and without your neighbor helping you? Because you would take your test all by yourself. So how would you approach this? And I'm going to pause this for a minute. Remind me to restart it. Okay, welcome back. We're going to hit this question in one more minute. But we talked about why you would use cDNA, complementary DNA. We just kind of jumped out for a minute to answer a question. And the reason is, prokaryotic cells and eukaryotic cells are different, right? They don't have the same control mechanisms. They don't have the same DNA structure, because eukaryotic cells and eukaryotic DNA has those little spaces of DNA, little spacers. They used to call it genetic junk. It could be failed attempts at evolution. It could be control regions, actually. But what do we call those regions that we cut out? Introns, and then we splice together the exons, right? So prokaryotic cells don't have that. So if you take a human or a eukaryotic DNA section and you splice it into a plasmid, which is one of your two types of vectors, right? What's your other vector besides a plasmid? Virus, right? It won't work for you, because they would transcribe and translate the introns as well, because they don't have the signals in order to remove those. So you need to give them DNA that doesn't have the introns in it and only has the exons. So what is suggested is use mature mRNA transcripts, ones that have already been snurped out. So take the eukaryotic cells, use its good mRNA that's ready to leave the nucleus and ready to get translated. Use that mRNA and use what enzyme? Reverse transcriptase, to make DNA that is absent. So you're working backwards, reverse engineering DNA that doesn't have any introns in it. That only has exons. And use that DNA to splice into your plasmid or your viral vector. All right? Okay, so let's talk about this one. So what is normal length wings is dominant over the allele for vestigial wings. So in a population of 1,000 individuals, 360 show the recessive phenotype. What does that 360 represent? Yes, and to get Q squared, that would be 360 over what? 1,000 individuals equals your Q squared, right? So what percent is that? 36%. So it equals 0.36, right? And that is your Q squared.
So you
Yes.
don't have to square it again. Like 360 over 1,000 doesn't equal Q, and then you square that.
No, it doesn't equal Q because I told you it's the phenotype. A phenotype is coded by 2 alleles, so it is your Q squared. 360 of 1,000, so 36% show the recessive phenotype. Do you understand that part? 360 of 1,000, so that's 36% of them show the recessive phenotype. So now I know Q squared equals 0.36. What's my next step? Square root, right? So I take the square root of this, I know that Q is going to equal what? 0.6. Once I know Q, what else do I know? P. When I'm dealing with, what is P going to be? It's really different. These are in my gene what? Pool. So now, and this is where your cautionary tale is, okay? Because it says how many individuals would you expect to be homozygous dominant and how many to be heterozygous? So how many would you expect to be heterozygous? Or let me do it easy. How many would you expect to be homozygous dominant? 160. Okay? You're not going to tell me a percent because I asked how many individuals. So if you were asking that, answering that on an AP exam and you had to bubble in your answer and you put 0.16, that would be wrong. That's saying, 16% of an individual, okay? I want to know how many individuals. It's 16% of 1,000 individuals, so that'd be 160 individuals, right? And to get my 2PQ, I do 4 times 6 is 24 times 2 is 48%, so that would be how many individuals would be a heterozygote? 480. Okay, does that help? Good? All right. So that, in essence, is chapter 16. I just wanted to hit that one time because we just finished that, and I didn't know if you had done your practice problems or not on that. Your next, it's 303. Chapter 13, your big ticket items on chapter 13, okay, is to understand how the expression of traits is controlled. So you want to be familiar with the operon in prokaryotes, and that's all about negative control, right? And then how is it different in eukaryotes? We're not so much about negative control, we're about positive control. And that's when you're going to need to talk about transcription factors and whether they are active or not. Okay, and remember we talk about activators being on the enhancer region, and then those activate the transcription factor, all of those things. So you want to review the parts of the operon. And it all, you can talk about positive and you can talk about negative control. And then when we talk about eukaryotes, we talked about the levels. Where's that control actually occurring? And when you just talk about controlling whether mRNA is made, that's transcriptional control, and this is where a lot of the control is. But before that, we talked about epigenetic inheritance, right? Things that control whether or not that DNA can even be transcribed to begin with. Is it tightly coiled and put away? Is there something above the DNA? Is there something controlling the DNA, whether that code can even be read so you can express it? That's epigenetic. Post-transcriptional control. If I have my RNA, what are some things I do to RNA once I have it? I cut it, I splice it, I cap it, I tail it. It has to leave a nucleus. That's post-transcriptional. Whether or not I'm on a ribosome or not, that is translational, right? Is some microRNA binding to it and inhibiting that? And then post-translational is, I have my protein, how do I need to fold it? What do I need to do for that? Okay, so that was chapter 13, and then we talked about mutations in there. And you want to review those mutations, right? There could be a point mutation. What's a point mutation? One single nucleotide. Okay? Are those always bad? No, they're not always bad because some of them are called, yeah, because they still code for the same amino acid. What's a really bad one we talked about? Frame shift. And that's going to be a result of an addition or a what? deletion, okay? Chapter 14 is all your biotechnology. What are the big ticket items in biotechnology? Okay, you've got to be able to cut DNA using what? Which cut a specific series of bases called the recognition sequence. You've got to be able to ligase and tape them back together. Cut and tape. Now, I need to have a way to get it in a cell, so what is that called? What are my two vectors? You already told them to me. Yeah. Now, and then we talked about how can I manipulate this? What kind of products can I get as a result of that? Organisms that have been altered with DNA from a different source are called what kind of organisms? Transgenic organisms, right? And we talked about other things we might want to do. I might want to get multiple copies of a DNA. You know, might want to run some tests on it. So I would do what? PCR. The polymerase chain reaction, that requires a machine, a thermocycler, which is like a DNA photocopy machine. We talked about how you will manually get it to unzip using a lot of heat, right? And then you have to use a DNA polymerase that can withstand the heat, so we're going to get that from Thermus aquaticus because it can handle it, right? And we talked about how you're going to have to give it nucleotides and primers in order to locate that. And we talked about genetically, we gave examples of modifying bacteria and plants and animals from that. We talked about gene therapy. You could do it within the organism, and it's called in vivo. Outside the organism is ex vivo. And then the last two kind of things were talking about genomics, and we talked about the Human Genome Project. We talked about the little sequences maybe being more critical and control than what we thought, right? We talked about when we look at, we can't just look at the DNA as the end all. We have to look at how things could interfere with the control, like microRNAs. We had to look at how proteins are folded, because that's your real picture. So we looked at proteomics. Do you remember that? And looking at proteins. Okay, so that was that. And using a computer, what did they call that? Biotechnology. Good. And then that leaves the final chapter of chapter 15, which is like a little history, a little bit about Darwin and his trip. Then we had a few scientists we learned about. That I think is pretty easy for you just to know those scientists. We talked about biogeographical evidence. We focused on natural selection, but we've talked about that several times. You should be really comfortable with natural selection at this point. And then we focused on evidence of evolution. And your big pieces of evidence of evolution, one is fossils. To be a fossil, how old do you have to be?
10,000 years.
That's right. And you have to be preserved, and the preservation process needs to not destroy you, and you have to be found by a person, right? We talked about anatomical evidence. There were 3 pieces of anatomical evidence. Do you remember what they are? Vestigial structures which point to your ancestral past, like a whale, hip, and femur, or little withered eyes in a salamander. What was another one? Embryological. How similar are embryos? More common ancestor. And what was the third one? Homologous structures. If I have a humerus, ulnus, radial, carpals, metacarpals, phalanges, and we all do, even though we're a cat and a whale and a sloth and a bat, we're probably related. We looked at did we look at the geologic time scale? Paleozoic, Cenozoic? No, okay, skip that. Did we look at biochemistry as a piece of evidence yet? No. Okay, so that's the big one, right? Because you're comparing DNA sequences, and you're comparing sequences of amino acids and proteins, right? And then we talked about, you can look at developmental biology, and we're going to hit that more later. Yeah. Okay, you guys, yes, that was it. Yes.
So homologous pairs, is it similar structure but not function, or is it similar function but not similar?
Okay, now, when you talk about homologous pairs, you're talking about homologous pairs of chromosomes.
Oh, I meant like
homologous structures is same anatomy, similar anatomy, so probably similar DNA. Analogous is just same functions. That's not a piece of evidence. Because lots of things have wings. It doesn't mean they're all constructed in the same fashion and related. They just happen to have wings or fins or whatever it is. They're handling their environment the same way.
So it might have different functions, but it's the same like structure.
Yeah, the critical piece is same structure means similar DNA, probably a common ancestor. Yes?
In chapter 14,
Wait, I can't hear. Shh, go.
in chapter 14, the summary talks
about nucleic acid hybrid nucleoside.
Oh yeah, let's talk about that.
So what is it?
Okay, let's see if we, no, that's great. Let's think what that would be. Nucleic acid, so I'm dealing probably with DNA. If I'm making a hybrid, you know what hybrids are, right? So, take my DNA, make it single-stranded. I could cut it up, I could amplify it, or whatever I want to do. Make it single-stranded, and I can look to see how much another organism's DNA matches my DNA. Or I could make it where I have known DNA that's a cancerous DNA of some sort and cut up my DNA and see if it matches it, because if it does, I'm probably going to get that cancer. So when you make a hybrid, it's two different, it doesn't have to be two different species. It could even be the same species, but you're comparing your hybrid, you're saying how similar is our DNA by seeing if it's complementary to each other. The more complementary it is, the more closely related, and the more likely you have that particular trait. Does that make sense? Yeah, because if you make it make sense, and it's not memorization, right? Anything else? Okay, so we'll have some time on Friday before you take it if you have some last minute questions that you want to ask me, okay? All right, you guys, make good choices. You're welcome, sweetheart.